Hello everyone and welcome to QuickMed, where medicine is explained quickly and easily. Today we will be discussing thyroid hormone production and walk through some examples of what happens when there are disruptions in thyroid hormone synthesis, particularly the up and down arrow questions that you often see on those USMLE test questions. So let's get to it. When we mention thyroid hormone, we're really referring to T3 and T4, which by their longer names are known as triiodothyronine and thyroxine, respectively. T3 is given the number 3 in that it contains 3 iodine atoms, and T4 contains 4 iodine atoms. So keep this in mind as we talk about the next few slides. To understand thyroid hormone production, we need to understand the hypothalamic pituitary thyroid axis, which regulates all of thyroid hormone production. It all starts with the hypothalamus, which produces your thyrotropin-releasing hormone, also known as TRH. TRH then stimulates the anterior pituitary to produce thyroid-stimulating hormone, also known as TSH. It is TSH that pretty much drives all of thyroid hormone production, leading the thyroid to produce T3 and T4. And once we've produced enough thyroid hormone, there has to be a feedback mechanism to inhibit production of thyroid hormone synthesis, and this happens by negative inhibition of the hypothalamus as well as the anterior pituitary. Now, if we take a look at the thyroid gland and actually take a cross-section through it, you'll find that the thyroid gland consists of these hollowed spheres surrounded by follicular cells, which you can see in the image there inside that red circle. And inside those hollow spheres, also known as the lumen of the thyroid, you'll find something called colloid, which consists of precursor proteins called thyroglobulin, which are key in thyroid hormone synthesis, and we'll talk about this in the next few slides. Let's now take a close-up look at those follicular cells as well as the lumen that they surround in this diagram here. On the left-hand side of this slide, you can see the basolateral side, which is where the blood vessels that supply the follicular cells reside. And then on the right-hand side of our follicular cell, we have the apical side, which refers to where the lumen is, and this is where all thyroid hormone synthesis takes place. Thyroid hormone begins with the uptake of iodide by the follicular cells, in which it is transported into the lumen, and then through an oxidation process, this iodide is converted to iodine. The next step in this process is known as iodination, and this is when iodine is bound to the tyrosine residues that are found on the thyroglobulin. Thyroglobulin is pretty much this precursor protein that is key to thyroid hormone synthesis. It's sort of an anchor that holds onto these iodine atoms. And when iodination happens, you produce two different molecules, MIT and DIT, and you can see their longer names listed there. And as the name implies, mono stands for the one iodine atom that is attached, and di is for the two iodine atoms that are attached. If you notice the star that is listed on this slide, it says TPO, and that stands for thyroid peroxidase, which is an enzyme that is actually responsible for iodination as well as the next step in thyroid hormone synthesis. Make sure to keep this enzyme in mind because it is going to be important to remember when we talk about some of the pathology that occurs in the thyroid gland. All right, now that we have our MIT and DIT molecules, TPO, or thyroid peroxidase, again performs the next step, which is known as coupling. Coupling involves binding MIT and DIT in various ways. When one MIT combines with one DIT, you get T3, and when two DITs combine together, you produce T4. And once T3 and T4 are produced, they are secreted into the bloodstream where they can act on peripheral tissue. Now, the difference between T3 and T4 is that T3 is generally considered the active form of thyroid hormone in that it acts on peripheral tissue. On the other hand, T4 is either bound to thyroxine-binding globulin in the bloodstream, or it undergoes a process known as deiodination in order to produce T3 where it can be active. Let's now go over some key points that you need to understand about thyroid hormone function. T4 is only produced in the thyroid gland as we discussed, while T3 is actually predominantly produced peripherally, and that is through the deiodination process that we just mentioned. And given that free T3 is 10 times more potent than free T4, it is generally considered the active form of thyroid hormone. And when we say free T3 or free T4, we're talking about the thyroid hormone that is not bound to any protein. Bound T3 or T4 is also known as inactive hormone. Aside from these key points, we also need to discuss some key structures in thyroid hormone function. We've already discussed thyroglobulin, which is the structure which houses the tyrosine residues upon which the iodine atoms attach to produce our different T3, T4 molecules. Thyroid peroxidase, as we mentioned, is a key enzyme in thyroid hormone synthesis. It's responsible for both the iodination as well as the coupling that we discussed earlier. And the last thing to remember is our TSH receptor. As you remember, TSH is produced by the anterior pituitary and stimulates the thyroid gland to produce thyroid hormone. It's important to remember the TSH receptor on which the TSH acts because there are different antibodies that can be produced against these receptors in the different pathology that we will see in the thyroid gland. Some of these antibodies can be stimulatory, some can be inhibitory, and so we will discuss this further in another lesson. Let's now go over some examples of what happens when this hypothalamic pituitary thyroid axis goes awry and what happens to the components that are involved in this. These make for great test questions on the USMLE or other types of medical school exams because they can ask about up and down arrows and what happens to what, and so let's go over these to better solidify our understanding. Let's now go over a case of primary hyperthyroidism, and this is when the thyroid gland itself is producing too much thyroid hormone for whatever reason. So in this case, we said the primary driver is excess thyroid hormone, so your T4 and your T3 are going to be increased. As a result of this, the T3 and T4 are going to have a negative feedback effect on the anterior pituitary and the hypothalamus. So this actually causes our TSH to decrease in response. And now you might be wondering, what about iodine uptake? Well, as we discussed, thyroid hormone synthesis starts off with iodine being taken up into the thyroid gland. And so in order to produce thyroid hormone, you need to take up a lot of iodine, and so that will be increased. Now on to another example, secondary hyperthyroidism. And when we mention secondary hyperthyroidism, this indicates that there is an issue in the anterior pituitary itself. That is, the anterior pituitary is driving this excess production of thyroid hormone. So again, as with the last example, our T4 and our T3 will be increased because we have excess thyroid hormone. And because we're producing so much thyroid hormone, we're taking up a lot of iodine, and so that's going to be increased as well. Now, because the issue is in the anterior pituitary itself, the anterior pituitary is stimulating thyroid production by producing excess TSH in this case. So TSH is actually going to be increased here rather than decreased, as we saw in our last example. Now for our third example, let's change things up a little bit and talk about exogenous T4 intake. So this is where a person is taking excess thyroid hormone through pills or supplements or whatever they may be. So this is an instance in which you have excess thyroid hormone, but it's not being produced by the thyroid gland itself. It's being taken up exogenously. So in this case, a person's taking excess T4. As we said, that T4 can be broken down to T3 or converted to T3, I should say, or remain as T4. So you'll have an elevation in both T4 and T3. As a result of this excess T4 and T3, remember this negative inhibitory effect on the hypothalamus and the anterior pituitary. So as a result, TSH is going to go down. Now what's important to remember is that TSH from the anterior pituitary is what's driving the thyroid gland to start producing some thyroid hormone. And so if the anterior pituitary is being negatively inhibited by this excess thyroid hormone, TSH is going to go down, and so the thyroid is actually not going to produce any more thyroid hormone, and so your iodine uptake decreases. Hopefully that made sense, but if it didn't, please feel free to watch the video again until you make sure you understand this because it's very important too. And for our final example, let's talk about another case in which there is exogenous intake, but this time it's of T3 instead of T4. Now, because T3 is being taken up exogenously, that's going to be elevated. And again, you're going to have this negative inhibitory effect on the hypothalamus and the anterior pituitary. So as before, TSH production goes down. This leads to decreased iodine uptake by the thyroid gland. And T4 here is also decreased because you're no longer producing any thyroid hormone. And as we mentioned, T4 is only produced in the thyroid gland, so there's no way to convert T3 to T4 in this case. So it would just be an elevation in T3 solely because of exogenous intake and not because of thyroid gland production. All right, everyone, I hope you found this helpful. This is a high-yield topic, so make sure you understand this well. Leave any questions you have down below, and please subscribe to our channel so that we can continue to make videos. And as always, good luck studying, everyone.